Well, good afternoon, everyone. Welcome to this WIN Distinguished Lecture. It's an honor and a pleasure to introduce Professor Mervyn Miles as a distinguished lecturer for this month. Professor Miles is Professor of Physics at the University of Bristol, where he is head of the Nanophysics and Soft Matter Research Group. He graduated from the University of Birmingham in the UK with a BSc in Physics, an MSc in the Physics of Solids, and a PhD in polymers from the Department of Physical Metallurgy. The Nanophysics Group has a worldwide reputation for the development and application of scanning probe microscopy, that's SPM, that is instrumentation for biomolecular systems. The group has invented new types of microscopy of particular importance in nanoscience and technology through its ability not only to image and follow processes at the nanoscale in most environments, but also to manipulate and write structures at the nanometer level. Professor Miles is also director of the University of Bristol's Center for Nanoscience, and is co-director of the Interdisciplinary Research Center for Nanotechnology, which is a joint effort of the University of Cambridge, University College London, and the University of Bristol. This center has been funded with £10 million over 6 years, and has also attracted considerable other funding, other investment. New buildings to house the activities of the IRC have been built at each of these three institutions, and of course, I think each one of those is slightly smaller than this, but nevertheless new. Interdisciplinary research is a key aspect of nanotechnology and is central to the philosophy of the IRC. Dr. Miles has worked in the USA, in Germany, in Japan, and the UK. He's a fellow of the Royal Society London, a fellow of the Institute of Physics, and he has published over 200 research papers. He was also a founding chair of Infinitesimal Limited, a spin-off from his research group, launched in July 2001, and now a successful company. In his talk entitled Touching and Feeling the Nano World, he will describe new developments in high-speed atomic force microscopy and shear force microscopy, which address limitations in conventional atomic force microscopy. Please welcome Professor Miles, Professor Mervyn Miles.
Thank you very much, Arthur. Thank you. So good afternoon, and it is a pleasure and an honor to give this distinguished lecture. So my title, Touching and Feeling the Nano World, well, you've probably guessed what the touching part is, which is using atomic force microscopy, which builds up images by touching. I'm going to leave you to guess what the feeling part is until towards the end. So this is our miniature nanoscience building compared to your building here. I'm very envious. I've really enjoyed my visit so far. A lot of very interesting, exciting work going on. I hope we can have some collaborations. So our building has been open for 2 years, and it does two functions. One, it's not a department. It's a center where people come and do research that need special characteristics of the building. So it's a center where people need to have very low vibrations and very low noise levels. It's also a cultural center in the sense that we want to mix all the disciplines from mathematics through to medicine. And we have, for that purpose, we have a nice coffee room. Coffee is free, and so people will come and perhaps talk. So lots of ways that we want people to mix and have new ideas and new directions, because we believe also that that's where the great developments will come. So I'm going to talk about atomic force microscopy. I'm a big fan of atomic force microscopy for the last 20 years or so. And for these reasons, yeah, it works primarily for biology. It works in liquid. Yeah? It works just as well in liquid as it does in air or vacuum. And we get three-dimensional, maybe 2.5-dimensional, because we can't see the sides and we can't see underneath. But we'll address that later. And you don't need to prepare the sample, so the biological sample is still functional and active, so you're able to make things happen and make things change with the sample. And of course, thinking of electron microscopy, there's no radiation damage, but of course we apply forces, and that's one of the problems that we have to overcome. We can measure other properties, in particular we're interested in mechanical properties, optical properties, electronic properties at the same time. And we can modify the surface, so we can start to write structures on the surface. And finally, we use the techniques we developed for the atomic force microscope for other applications. In particular, we're developing at the moment sensors. So it's nothing particularly new. I mean, that's just been around for probably 15 years using microcantilever sensors to detect biological molecules. But what we've learned from our high-speed atomic force microscopy now gives us greater sensitivity to operate even in liquids. So I'm not going to talk about those today, but that's a spin-off from what we're doing. So that was the good news. The bad news is that the imaging is very slow. If you've ever done atomic force microscopy, you know it takes a minute or so, maybe up to 5 or even 10 minutes, depending on the sample, to get one image. And so what we need to do is go faster. We actually like to be going at sub-millisecond per image, if we could, in order to capture some biological processes. The second thing is there's no low magnification. Yeah? In AFM, we have to take a window and scan it round. Unlike electron microscopy or optical microscopy, where you can zoom back and get the whole sample in, we can't do that with atomic force microscopy. We're stuck with about 100 microns square. And so if we could go faster, if we could push the speed up, we could start taking lots of images quickly and building up a large area. So the speed improvements are important. Secondly, we don't really want to have a force that distorts the sample or biases a process. So we want to put as low a force as possible, and we've addressed that in 2 ways. So I'll mention that. And finally, something that we've actually, as SPMers, we've almost forgotten, and that is our samples are nearly always planar. In fact, we don't even think about not imaging a planar sample. And so that's a clear restriction, because life isn't planar, life isn't two-dimensional. So how do we look around a 3-dimensional object? Okay, and that will come towards the end. So I'm going to talk about high speed in contact mode. You say, surely that's going to cause a lot of damage. High speed in shear force or transverse dynamic force, because we don't just do shear force, we do other fields as well. And that's a non-contact technique. And then I'm going to talk about optical AFM, which is using holographic optical tweezers to control the AFM probe. So first, the high-speed contact mode. This is how we started. So we started actually doing high-speed near-field optics. So where you oscillate an optical fiber, you coat the fiber with aluminium, have a small aperture, you bring it into the near field, very close to the sample, and you get resolution beyond the diffraction limit. What we were doing there was controlling the distance by this shear force, oscillating our fiber, feeling the surface before we touch it, actually through layers of water molecules. And then we invented a high-speed version of that, where we, instead of just dithering this little probe by a nanometer, we actually whacked it up to microns and just collected the data. And we were able to get 100 frames per second compared to 1 frame every 20 minutes. So that was a useful breakthrough. And then I said one day, why don't we just try doing AFM? This was a stupid idea. So we took our tuning fork that was driving our optical fiber, and we turned it on its side, and we put a sample on it. In fact, we put 2 little glass blocks. These are only a millimeter square. This is a watch tuning fork. It runs at 2 to the 15 hertz. It runs at about 30 kilohertz when you've got some mass on the end. And we used an ordinary AFM cantilever, and we did the usual beam deflection to measure the bending of the cantilever. And we imaged a polymer sample. We clearly knew that the sample was going to be destroyed by doing this, putting a sharp probe with a high force, 100 times higher force than normal, in order to accelerate over the bumps. Scanning it at 30,000 lines per second compared to 1. The sample's not going to survive, but it was like, let's just see how bad it is. The sample's going to end up on the wall. But after 20 minutes of scanning, we found we'd done no damage at all to the soft polymer sample. So that was a silly thing to do, yeah? But it meant that we had a breakthrough. It meant that we could image, for example, here, this is on the right-hand side, quite fast normal AFM, and on the left, video rate using this. So you see this is just flickering. This is video rate AFM on the left, and this is normal rate on the right. Same sample. Looks a little different, but that's one of the very early images from the microscope from our spin-out company. So that little tuning fork was a pain, yeah? Trying to put the sample onto this micromillimeter size little block and counterbalancing. Trying to make that work in liquid was a pain. So we built our own stage. So now instead of being a millimeter in diameter, we've now got a centimeter in diameter stage. And this stage actually goes faster than the tuning fork. We can now do 100,000 lines per second. And we can go over about a micron. And this fits on a Vico or Brooker dimension. Here's the liquid cell. And here's how it looks. This is Lauren Pico, who's been the postdoc at the heart of the high-speed AFM for the last few years. And apparently on the screen behind, he is looking at some of the early images. So this is collagen. You can see the banding pattern. You can see the microfibrils. Very early images. And what we're doing is scanning at video rate and then just panning the tube back and forth at the normal speed that you would scan. So we're getting video rate images that we then raster scan down the page. So here's an optical microscope of some collagen fibers. And here is the same image as was in the square here, taken with our latest version of the high speed. Okay, this is not one image, this is 4,000 images that have been tiled together. You can probably just see the banding, they're not stitched perfectly together. And so as we just look at a few of those, you start to see the structure. So here you can see the tiled pattern underneath. So now we have a way with a closed loop stage to scan very large areas up to a centimeter and collect data at about 2 nanometers resolution. So that opens up a lot of possibilities in material science. And then Lauren and another student, Ollie Payton, came up with the idea of using contact resonance at high speed. So I won't go into detail what that is, but if you use contact resonance, it means you can get the stiffness out of the material in contact. And so here's the collagen again. Here's the topographic image. High is white, low is dark. But here's the mechanical property at the same time. So you can see that the collagen is softer than the silicon substrate. So we're able to map in 17 milliseconds not only the image, but also the mechanical properties. Now, if you've ever done force volume with AFM to try and get the mechanical properties, you know you end up with a highly pixelated image after about half an hour. Okay, and it works in liquid. Yeah? So far it's been in air. Here are some human chromosomes taken with conventional tapping mode AFM. Then we've hydrated them in buffer, and they've swollen by a factor of 5 from the dry down state. So now we've got a huge structure. Yeah? So if we're just scanning this tip over the surface, surely when it hits half a micron high, soft, gel-like chromosome, it's going to damage it. So here we go. This is real time. We're just moving around the chromosomes, chromosome 22 there. And so it's a bit like an optical microscope. Yeah, you can just move around in real time. So you're probably saying, well, I can't see any detail on that. Well, if you sit on one of the arms, you can start to see things at the kind of protein level. So you can. So this thing is working even in liquid on a soft sample. Okay, one more example. This is dental enamel. This is adding orange juice, essentially. The right-hand one has been, dental enamel has been soaked in fluoride, and the left-hand one not. So let's look at the difference. So here, a few pieces are coming off as we add the acid, but not too bad. And now look what happens when we add it to the untreated tooth. So here's a health warning, yeah? Look at all those crystals being dissolved. And of course, we're going down several, hundreds of nanometers, maybe a micron even, in that dissolution of the enamel. So we wondered just how fast we could go, so we can do a video rate, but how fast could we really push this? And so for the anniversary of the invention of the AFM, we thought 1,000 images a second would make a good headline. We did a quick calculation and we thought we could do it. Now we have to use our high-speed stage for the slow scan, for the frame rate, because 1,000 frames per second is faster than a normal scan tube will scan even a line. And now to do the high speed, we use another tuning fork that's 100 kilohertz. So we're doing 100,000 lines per second. And it still works. It's not pretty, but this is 1,200 frames per second of the collagen again, obviously slowed down. So if we have to, we know we can go sub-millisecond. So how does this work? Why aren't we destroying the sample? The clue came very early on. This was actually in Lawrence's thesis. We measured at low speeds the friction force, the lateral force. And then as we wound the speed up, there was a transition. It went from this kind of square wave to this sine wave. And it's a sine wave because we're moving the stage sinusoidally to avoid resonances at turnaround. And so what you're seeing here is something that depends on velocity, force that depends on velocity. So there's a clue. That's a viscous effect. So what our hypothesis was that the tip, when we went fast enough, lost contact with the surface and lifted off the surface and felt only the viscous forces as it moved through the liquid. And Ollie did a lot of work measuring. This is real data. This is taken with a scanning laser vibrometer. So we can measure the behavior of the cantilever at all these points. You can see these dots are all the points it's been measured at, all the different modes as we go over a step. So we've analysed what's going on in the cantilever, and we've measured its height, and we believe this is what's happening, that as the probe moves through the liquid, we build up a hydrostatic pressure on either side, and that lifts the tip off the surface, which lowers the pressure, and so we have an equilibrium position of the tip above the surface. Here's some calculations. So we're treating here's our tip, here's our surface, here's a water layer. Now this could be an air, and it could just simply be that water layer and the water layer on the tip. There's always tens of nanometers of water on things under ambient conditions. But it could also be the water in a bulk water, because near the surface, the water is ordered, and it has a different mechanical property, much higher viscosity, much higher elasticity than the water in the bulk. So we have two viscosities, one for the bulk and one for this water near the surface. So taking that it's not really an assumption because we've measured it, we can build models, and here are different models of if we change the viscosity. So if we increase the speed, supposing our viscosity is 0.3, at some velocity, about 0.5 millimeters per second, we'll undergo a transition where the viscosity suddenly goes down. So we come here and it suddenly drops. Over here you've got the friction, I think. Again, if you go to a reasonably high speed, it's pretty high speed, 1 millimeter per second, there's a transition as you cross that. And you can then vary other parameters, the tip radius, the load you apply. And what you're seeing is that about a millimeter per second, there's a transition. And what's happening is the tip is lifting off the surface. And here's the data directly. So this is the experimental data. So the tip is only lifting off a nanometer, so this took a long time to be able to record. The thing is moving at up to 5 millimeters per second there. And we get this discontinuous jump. It doesn't just move off gently, it jumps. And if you look at the theory, it's bistable. When you get to this point, it will jump because of this bistability in the plot. So it's not matching exactly, but you can see essentially it should go here and then jump and then continue. So the tip is lifting off the surface if you go fast enough. So that's why it worked. We went fast enough just by chance, and we got that result. And then you can look at the friction, similarly at about 1 millimeter per second. Again, a bistability in the friction. So you mustn't go too slowly. Okay, now here's a little bit of fun. This has no real use. Yeah, two things that are fun to do, but not very useful. This is, what can you do if you've got video rate? What's different? Well, you can make a different interface. Yeah? So all normal AFM, you watch the line scanning, you set the parameters, the scan speed, the feedback loop, and so on, and it captures the data, and you can go and have a cup of coffee or whatever, and come back later. But if you've got a video rate, you can interact with that instead. Yeah, you can actually use, in this case, a multi-touch table to control. So we built a big iPhone, and now we can drag the scan area around with our finger. Yeah. So this is the area that's being scanned, and this is them being stitched together. This is just a calibration grid. And then you can do the pinch to change the magnification. So it's a very different interface. You don't have to be an AFM expert to use this. And you can do translations, and I'll let you do the translation now. That's not a pipe. And then here is a use for it. This is tracing around a chromosome, one of those human chromosomes. So on the left, we're just keeping the interface. So this bit is bright and this bit is dark. We're just at the edge of the chromosome. So what we're doing is tracing around, making a mask so that we can set different parameters inside and outside. So we've got the hard surface it's sitting on, but we might want to change the feedback and the forces we're applying on the inside where the chromosome is a lot softer. So we can draw around it and make a mask where we automatically change the settings. And now here's really something that's completely useless. Yeah? Wouldn't it be fun? So we've been oxidizing silicon by putting a minus 10 volts on the tip in a humid environment. So the oxide takes up more volume, so you get a little structure on the surface. And we did that first just with a spreadsheet, yeah, and just normal speed, wrote some dots about 20 nanometers across. Oxide's great because you can then reduce it and deposit other things in those locations. But wouldn't it be fun just to do it with your finger? So put your finger down, there's the oxide formed, and now drag your finger around. Okay, it's a bit noisy because we're writing and reading at the same time, and the conditions for those two are not the same. But that's what we drew, some cave painting. Definitely a Friday afternoon experiment. Okay, so the second thing I want to talk about is also high speed, not anywhere near as high speed as the first bit. But the point is here, it's non-contact. Yeah? So the tip doesn't actually touch the surface. Well, it didn't in the other case either in the end. But here we start off with it not touching. So this is more appropriate not for large structures high with great height differences. This is more appropriate for the kind of molecular level. So this is an AFM in which we have the tip mounted vertically. So it's very like our near-field optics, yeah? Our vertical optical fiber. And we do dither it. We move it by about a nanometer at the end, and we measure its behavior with a beam deflection system again. So that's the original version. That's how it looks in reality. Here's the sample. Here's the scan tube. Here's the probe. And then we have two optical detection systems, because now it's circularly symmetric, so the probe might start to do Lissajous figures, and we need to know what it's doing. And the kind of thing you can get out, well, first as you approach the surface. It's going to stop there, not. Just putting that back to there again. Okay, so the first thing, this is the amplitude of oscillation. Probe is oscillating, and we're measuring the amplitude. We're approaching the surface, so here we're 2 nanometers, and then something starts to happen. The amplitude goes down to about 1 nanometer. But look, there are peaks. Yeah? There are peaks in this, about 2.5 angstroms apart. First it returns to the same level, and then it starts to go down. So these are the water layers. Yeah? We're confining the water between the end of the tip and the surface, and the water starts to order. And when it orders, it has a different mechanical property. Yeah? So the viscosity goes up and the stiffness goes up. So that's what we're detecting. we're feeling the surface, not by putting a normal force on, but by just shearing a little bit and feeling how the water property, mechanical properties have changed. So we know where the surface is without touching it. What you can also do is do that in 3 dimensions. Okay, so now you can also do that at every point. So you're oscillating the probe and you just move it in and out a nanometer at every pixel. So you can map out the water layers across the surface. So probably quite important for biology to know how the water structure is on different regions of a membrane or different parts of a biological structure where you'd want ordered and disordered water. So here's the kind of images you can get out. This is DNA. This is one image, of course, just being looked at in different directions. You can just about see the double helix, and that's in air. Okay, so let's go faster. So the way we're going faster in this case is very similar to what you can almost call the conventional high speed, where you just make everything on a shorter time scale. You just make your cantilever smaller with a higher resonant frequency, you just scan everything faster. You just change the time scale of conventional AFM. So we'll do the same with this. We make the cantilever smaller so it has a higher resonant frequency. And then, of course, we're looking at each pixel, so we still need to do feedback at each pixel. But the great thing about this compared to ordinary AFM is this is a vertical cantilever. It's essentially infinitely rigid in the vertical direction. So it doesn't get pulled onto the sample by capillary forces, by van der Waals forces. It's also, of course, non-contact, and the probe can be made from anything. Yeah, as long as it resonates, the probe can be made from anything. It's circularly symmetric, so you can start looking at properties, directional properties. And practical importance is the carrier of the probe is away up here. It's not very close to the probe tip as in conventional AFM. So here's a probe. Here's a 7 micron long probe, 3 megahertz in our microscope. You can just about see the end shining. So the end is shining because it's sitting in an evanescent field. So we create an evanescent field on the surface. We dip the probe in. The probe diffracts and scatters. We collect the scattered light, and by looking at the position of the scattered light on the detector, we can measure the position of the probe to about an angstrom. So that's the diagram, essentially the same. That's how it looks. It looks homemade. And here's some rather old set of images, but this is DNA again. We can do about 5 frames a second at the moment. So it's mostly a control engineering problem now. So we're working with our control engineers to improve the feedback, because that's where our weakness is in this system. There's another mode you can use it in. We've got that evanescent field, so we know when the probe dips into the field, it'll scatter, and scattered intensity decays exponentially with distance, as is shown here. So as we move further away, the field, the scattered light decreases. Yep. So this is scattered intensity. So at a given level, we have a given intensity of light. So that means we can fly across the surface at high speed without any feedback, except for the optical feedback. Just keep the intensity of light the same. So you're always scanning parallel to the plane. And then we can measure the mechanical properties. We can measure the mechanical interaction between the probe and the sample. Scan really fast and just see what the coupling is to the sample. So this shows us approaching the surface. This is away, and then the red bit just shows where we are, just clipping, just interacting, not really clipping, interacting with the tops of the DNA molecules. And you can see the DNA molecules emerging from the background, and you see here a lot of more background noise from the surface contamination. But here you're about right, just seeing the DNA molecules. So we can do this really fast because there's really no feedback involved, which is the limiting. So now we have a high speed non-contact version. Okay. So another thing we can do with this technique is to measure very small forces. And this is really Massimo Antoniozzi's work. So it's the same system. We've now made a very, very soft cantilever, very low spring constant, attached to kinesin molecules, this is a motor protein, and brought it up until it's in contact with a microtubule, and then let it progress along the microtubule. This is the cantilever, perhaps one of the softest cantilevers anybody's ever made. But remember, we're using it vertically. So if we didn't use that horizontally, we'd just stick to the surface again. We're using it vertically. So now we're going to measure a force. And so as we can measure force here, this measuring displacements, we're just letting the molecule walk along, letting the kinesin literally walk along the microtubule. And you can see the steps. Yeah, you can see the 8 nanometer steps in this data. And then at a certain point, it stalls at about, when the force gets to about 5 piconewtons, it stalls because now it can't move against the force of the cantilever spring and jumps back. People just thought it just came off and jumped back, but when you look at the detail in there, you can see there's a lot more information. It actually is like running backwards. Okay, so I've shown 2 types of high-speed microscope. One, large areas, large specimens, large height differences, very high speeds. And the other one, more appropriate for single molecules, low forces, water structure information, and very easy probe fabrication of making probes of different types that might even interact with the samples. There's a lot of different modes, it's a very rich area, which I'm not going to go further with, but because I want to talk about the final part. So this is the team from the high speed. Dave Ingledew is our technician who builds the microscopes. Massimo, I mentioned, Lauren at the center, 2 PhD students who've been working on the 2 types of microscope. Arturus wrote a lot of the software. And Pete is the biological person that keeps us doing the right things. So the last part of what I want to talk about is the holographic assembler. This is a holographic optical tweezer system. So we're going to talk about not scanning in a plane anymore. Okay, so that's the optical tweezers. Okay, so this is the artistic impression, that you can use the laser beams to grab things and move them around. So optical tweezers, invented in the same year, coincidentally, as AFM, 1986. So it works by focusing a laser beam down using a lens, objective lens, the optical microscope. You create this waste. And you can think of it in 2 ways. If you think of it in a kind of macroscopic way, you know that light refracts at the interface between different refractive indices. So if this is a glass bead, it would bend towards the normal, and then as it leaves, it bends away from it. So there is a change in direction of the light, and the same on the other side. And when you get a change in direction, if there's momentum associated with it, there must be a force. So, and if you do the analysis, the force tends to keep the particle on the axis of the beam here. So if it moves off, there's a force to move it back. If the particle is not on the axis, there must be a force. That's the sort of simple way of thinking about it. So instead of just the lens, we've put a spatial light modulator here, a dynamic hologram here. And so the beam is expanded onto this hologram and then refocused back into our sample in the optical microscope up here. So what are we going to do with a hologram? We're going to put diffraction gratings on to split the beam up into multiple beams. And we're going to put Fresnel lenses on to change the focal length. So that means we can make several beams, and it means we can change the focus in Z, in distance. So instead of all foci being in the same plane, we can now move them around in 3 dimensions. That's how that looks. It's a big project. Enough said. So here's the first interface. So what's happening? That's a real hand on the left with a glove with spheres on the tips of fingers and thumb. We can calculate the X and Y position of these, and the Z by the size of the sphere. So we get three-dimensional coordinates of those fingertips. So in real time, we calculate the holograms that you would need to focus the laser beam in the optical microscope to the same positions. So over here, where the red circles are, is where the laser beam has been put by the hologram. And these are little spheres, micron-sized spheres. Let me run that again. These are micron-sized spheres that have been moved around. Yeah. So you can see whatever this hand does, this micro hand does the same. Yeah. So you can see also in Z, okay, you can see this one's gone out of focus. You can see the defocus of the ones that are in different planes. So the thumb is way down, so it's different. Yeah. So you've made a little micro hand. And here we're doing a different thing. We're moving something that you can't trap. This is a piece of chromium. So the laser would just kick the chromium out. You just get total reversal of the light. So all the momentum would kick the chromium out. But we can move it indirectly. Yeah, so you can trap, so this demonstrates you can trap indirectly. You don't have to put the laser onto the sample. And then for no reason at all, we're moving a white blood cell around with 3 red blood cells. One day there'll be a use for that. And we can't resist. We've got a multi-touch table. Yeah? So now wherever you touch the table, we calculate the hologram to put the laser beam in the same place as the image. This is the optical image from the microscope. You can see there are spheres on here. Wherever you touch, we calculate the hologram. So if you touch a sphere, the hologram goes to that point in the microscope, focus goes to that point in the microscope, and you can then move your finger and the sphere is moved. So here we are just clearing the table. Sweeping things off. And here we're playing around with cells. These were directly trapped, but still seem to work. Leukemia cell and the killer cell, bringing them together, holding them together for a while, taking them apart, and then leaving them longer times, and then they form some kind of process between the two things. So opportunities for looking at cell-cell interactions. And then we can make a little nano tool. So here's a nanorod, and we put our micron spheres on it. Now we can control our nanorod, yeah, in 3 dimensions, any orientation. So we could functionalize the end of the nanorod. So instead of having this complicated syringe that was proposed earlier, we can actually have some kind of coating to the end of this probe that would stimulate the cells in some way or break the membrane down. So to make that, we use our multi-touch table. We bring these spheres together. So here 2 people are building the probes. The spheres are now linked together. So you can write software that obviously can easily just move them both at once. And then we need to add the nanorod. So here you can see the Brownian motion of the nanorod until we trap it. And then we can bring the spheres and the nanorods together to make that probe. It's quite difficult, actually. And here's the advert. There's an app for this if you want to do this at home. No, seriously, there is. If you go to the App Store, there's a simulation of all this called iTweezers. It's free. And if you actually have a holographic tweezer system at home, you'll be able to. We then found that we didn't need to do all that by hand. We actually could automate it. So here, Dave Phillips managed just to pattern recognition, get these spheres at different heights, grab them all, bring them to one plane, then put them into a predetermined location. So here's a kind of AFM now. It's very crude. Here's spheres. You probably can only just see the nanorod. Can you see that? Just about see it here. And we're bumping up again. So what we're doing is holding the beads trapped and moving everything else. And the reason we want to do that, we want to be able to measure the forces. So when the beads are not sitting in the middle of the trap, there must be a force. So we're plotting the forces that the bead experience, that the beads. Just that again. See, you can just, it's a bit weak, you can see the forces along the axis and off axis. So the forces are quite low. The noise level is 50 femtonewtons. So we've kind of got an AFM probe. We're measuring force. But what we'd really like to do, we'd like to be able to look around a sample with AFM. We'd like to look at the sides. We'd like to look underneath even, yeah? And we can't do that. We'd really like to look at all those places at once. So we can't do that with AFM probes. It'd be a complete tangle of cantilevers. But we could do it with an optical trapped probe. So if we now trap the nanorod, and maybe we have several of them, we can move it around the specimen because we can control this. We have 6 degrees of freedom we can move it with. So we can move all the way around an object and we can scan it in different places. And we could have more than one. So artist impression. So we're not going to actually do all the things I've just said because we found out we don't need to. Yeah. So we bring the probe up to the sample, and when we touch, the probe doesn't move anymore. So we know it's now touched the surface. Sometimes it becomes skewed, not perpendicular to the surface anymore. So we know we've interacted. So all we're doing, instead of measuring the force directly, is measuring when the probe doesn't move where it should move. So we do it. What's happening in each of the images I'm going to show you, we do it crudely, these blue lines, these blue points. Whoops, we've hit it, then we back off, and then we do it more sensitively, more with higher resolution, and then locate the position where we first touch, and keep repeating that, or obviously automatically. And we found we don't actually need to put the spheres on. So here's the very first set of images. So they're a bit pixelated. But here's the probe being scanned by the holographic tweezers back and forth on the side of this structure. And here we're collecting forward and back. Not many points in the image at this stage, but I always think it looks better in 3D. So we're looking at the side, yeah? So we couldn't do that with AFM. And then we had this crazy idea. So the big problem with this is making the probes. Yeah, it takes a long time to find the nanorods and to find the spheres and to bring them together, to glue them together, to make the probe and then not lose it, yeah, by the time you want to image. So what you need is a way of making lots and lots of probes so you have like a bucket full of them. Yeah? So we turn to nature. This is a diatom, yeah? This is a living alga. So it's made of silica on the outside. Yeah? That's great. And it's got, conveniently, 2 oil bodies that are tethered in it of higher refractive index. So you can see where I'm going here. Yeah? You can use this as the probe and trap with these 2 points. And you've got, literally, you can grow these probes. And you've got to keep them alive, otherwise the silica cracks open. So you've got to be nice to them. See here we've arranged them into a nice... So we actually use this to do the imaging. So here are these 2 spheres. This gives us an idea of resolution. The resolution isn't great because nature hasn't been kind to us and made a really sharp 10 nanometer tip on the end of these. Yeah. Maybe with genetic engineering we could do that. But. So the first thing we looked at is this other alga. It's this Pseudopediastrum, which is star-like, as you would guess, and these cusp structures all around here. This is quite soft structure. This is a confocal fluorescent image of it. So what we're doing, we're bringing up that diatom, using its 2 oil bodies to trap it, moving it towards the surface, and when it touches and comes slightly not in the position it should be, according to how we've moved the traps, we detect the surface. Okay, so what's the resolution limited by? Well, obviously if you've got a blunt tip, it's limited by that. But even if you didn't have a blunt tip, at this level, you've got thermal motion, you've got Brownian motion, because this is not rigidly fastened to the rest of the microscope. It's sitting in optical traps that are fairly soft compared to a real mechanical connection to the microscope. So we need to do one more thing. We need to feed back in order to reduce the tip position. We need to know where it is and then correct all the time. So you can only do that at a certain bandwidth because you have to have the update of the holographic traps faster. Making the holograms, calculating the holograms is easy. The graphics card can make 400 holographic traps at 70 frames a second, thanks to the games industry. But the display can't. Display can only do 200 frames per second. And that's good, but that's due to 3D TV. You need the 200 hertz frames. So we can do 200 images a second, so we can correct the position. And that's what's happening. In the different orientation, to different degrees of freedom, you can see we turn this on, so you get in this Brownian motion, some high amplitude, low frequency stuff, and then we turn it on, and we've clamped it. Yeah? We're holding now the end. We've lost a little over half of the amplitude, RMS amplitude, by putting the clamps on in various modes. So that's what we need to do. There's our probe, about to image that. And here's one of these cusps imaged from the side. And there's two. But we found a better way of making probes. And if you haven't got one of these, you definitely should buy one. Yeah, I think a Nanocenter should have one of these. Two-Photon Polymerization, have you got one? No. It's wonderful. You can make three-dimensional structures at the moment with about 100 nanometers resolution. We've just discovered another polymer to polymerize, which gives us 60 nanometers resolution. So you can write in 3D with 60 nanometers resolution. So you're getting two-photon polymerization of this monomer. And you can make these structures. Okay, so you can make, so we do a lot of simulations as well, a lot of calculations on the electromagnetic field and the forces and the torques and the hydrodynamics. But we also calculate, if we want to make this end really stable, where should we be putting the spheres? Where should we be putting the forces? So different structures that we've tried. This is ideally what we want. We want the traps trapping the beads, and then this sharp point coming out. We could trap a nanotube in this stuff. Yeah? You could polymerize around a carbon nanotube and make it even sharper. So we've changed that system that I showed originally slightly. What we've added is a stereo optical system at the top in order that we can tell the Z position. Yeah? So we can get X and Y, but we need Z. And so this is a system where we're sending two different polarizations of light in and collecting the image, and then we get offsets depending on which polarization we're looking at. So we can tell the depth. Yeah. So if it's above a plane, it's one way around. If it's below the plane, it's the other way around. If it's in the plane, the images are coincident. So we can now get X, Y, and Z for each of those beads. And so we know its position, we know where we put the trap, so now we can measure the forces in 3 dimensions. And there's so many more things you can do. The more you think about this, the more interesting it gets. So with a spring, yeah, cantilever, AFM cantilever, if it's displaced by a lot, yeah, so you're scanning and suddenly you come to a high point, the spring bends a lot back, the force goes up. You didn't really want that, because that's going to cause damage. With this, you can create a system where the force stays constant. So here are the traps. We can move this thing around by working at the end, move this probe around. But as soon as it touches and this trap moves down this bar, the force doesn't change. Yeah. So we touch, but it's like it slides then. The force is constant. So we don't put extra forces on. And so we're imaging this side of the silicon structure. So here we bring the probe in. So you see the Brownian motion is higher up here. And then we start to image the surface with one of these probes. So I think this is, this is the direction we're going in. We can now trap, we're going to start now trapping 3 probes so we can scan in 3 orthogonal directions at once. And we've increased the resolution. And we've realized we don't need to care about the Brownian motion. It just gets better all the time, doesn't it? You don't need to care about that because as long as you know where the tip is, that's what you need to know, yeah? You know the tip is here, at any time, then you know where you made contact. And the fact it's wobbling around doesn't matter. You just know where it is, so you get the information about the point where the tip is. Okay, so I promised I would talk about the feely bit. We did the touching bit with the AFM, yeah, feeling. Now we're going to feel. And as I've said several times, if you take an object like this and you have the traps, if those traps are not coincident with the particle, there must be a force. So now we've got the force in 3 dimensions. We can feed that force back to a cyber glove. So now we can start to feel what those particles are feeling, if particles feel anything. Yeah. So here it is. So it's a bit of a complicated, it's called immersion cyber glove. I'd say, nope, not everybody is wearing cyber gloves today, so I can't let you feel what it's like. But here we're squeezing, can you just see this vesicle? We're squeezing a vesicle with our, and we actually burst it. So it's fun to do it. The idea was that you would do it in order to palpate a living cell, to feel if a cell was diseased or not, in the way a surgeon might palpate an organ, see if the internal structure is not what it should be. But it turns out, using these spheres, we don't actually have enough force. We don't put enough stress on to deform it enough. What you can do, though, is you can feel that you've touched it. So you can pick a cell up and you can turn it around. You've got that feeling that you've touched it. What we need to do is increase the force or increase the stress. So what we really need to do is probably take one of those probes with a sharp point, so the force is the same, but the stress is higher, so we can deform the membranes and measure higher deformation, feel the cytoskeleton inside the cell. But that's something we still need to do. So with that, I'd like to thank our collaborators in Glasgow, excellent collaboration for the last 8 years with Miles Padgett's group, helping us with some of the optical problems. And my own holographic tweezer group, Dave Carberry, has just left and gone back to Australia, but he was heading up the holographic tweezer operation. Simon Hanna and Steve are doing all the clever calculations, coming up with quite a lot of ideas from the calculations. James is making the probes with silicon fab. Dave Phillips is doing an awful lot with the AFM, optical AFM. Sri is our computer science guy who's been helping us with the cyber gloves and the multi-touch table, both of which he thinks are very old-fashioned compared to what you can do these days, but we don't really know what he's talking about most of the time. And then a couple of PhD students, there's quite a few more actually, this is a bit of an old photograph. And Daniel Robert, who together with me, we had the original idea for doing this over a glass of wine. And finally, to thank our funding agencies, including our spin-outs. And thank you very much for your attention.
I just wonder, for this high-speed AFM, so what is the finest spatial resolution that you can achieve?
In the case, well, it depends which one we're talking about. And it depends on the sample, as ever with AFM. So if we're talking about fairly rigid samples with a high-speed contact mode, it's about 2 nanometers.
2 nanometers. Oh, that's very good.
If you're talking about the non-contact, the vertical probe, it depends on the amplitude. At the moment, it depends on the amplitude of oscillation. So you can see DNA there, that's just 2 nanometers across. So something of the same. The vertical resolution is much better in both cases. Yeah. So we don't see atoms, but we do see, well, we see biological structures. We've got enough resolution for what we would like to do.
Yeah. Have you tried this conductive AFM high speed?
No, that's on the list of things because when you do the high speeds, you've got the mechanical properties, you've had the conduction as well. Yeah.
Right.
We've done the writing, of course. I mean, that's sort of conduction.
Right.
Yeah. So you could do something.
Because at high speed, you're actually floating the tip above the surface. Right?
Yes.
In that case, you are not really physical contact.
No, but we're within tunneling distance. And it's water, anyway. So
Right.
it's, yeah.
In one of your simulations, you show the viscosity of water.
Mm.
It changes from 100 to 300 times higher than about water.
As you yes,
Yes,
small.
it's a gas score.
Yeah.
Are you able to measure the viscosity using your AFM?
Yes. So you can work. So it's an inverse thing. You can work backwards, knowing what the forces. You can calculate what the forces must have been on the probe. So we've done, I mean, it's not very, I think we've published it, but we've got the complex, so you get the complex modulus out. And of course, the question is, what is the last layer of water? Do you ever really see that, or is that so ordered that it acts like a solid?
So it's structured by hydrogen bond, is that?
Yes, and then the confined geometry means that you can only fit an integer number of molecules in the gap. So you get the oscillations because you get something commensurate with the gap, then you move that boundary a bit, and now they won't pack, so it becomes viscous. And then you move a bit more, it becomes elastic again, and so on. Okay, yes, so that's another type of microscopy which we're always coming towards, yeah? That's photonic force microscopy. So you've got your potential well of the optical trap, and the bead that we have sits nicely, comfortably in that at about a micron. But if you make it smaller, then of course it can, with thermal energy, it can rattle around inside that. So if you do the same thing, you track where the bead is, then you can look at the potential that it feels. So that's a photonic force microscope, essentially. So we're doing the same thing, but now we're also scanning. We're saying we don't care about the thermal motion, we'll just follow the position of the tip. So we've come almost to the same place as originally, but what we can do is have multiple probes. The photonic force gets stuck when you have more than 2 probes because you can't track them very well.
You mentioned about the silicon nitride cantilever. You said this is the smallest one ever made.
Softest, yeah.
The softest one. How is it made?
It's a silicon nitride layer, silicon with 50 nanometers of silicon nitride, and it's just normal silicon fab etching. But okay, you have to be a bit careful. It's 200, 150, 200 microns long. I guess nobody really wanted to make one soft before, that's why. But because it's no use, in normal FM that'd be absolutely no use. It would just stick to the surface the whole time. You wouldn't be able to lift it off. But in our case, because we have it vertically, we can measure the lateral force. So yeah, so it's just silicon nitride etching and then removing the silicon.
What's the depth size?
Sorry, the
depth size.
Depth.
That is the size of it.
So it's I think it's 1 micron wide, 200 microns, 150, 200 microns long, depending which one, and 50 nanometers thick. So very, very thin. And the curious thing is that we thought when we dry these, when you take them out from the etching process, wet etching, that we would destroy them. But okay, they bend and they curl up, but as soon as you put them back in water again, they straighten out. So sometimes nature is on your side.
So you have one slide that introduces us the optical tweezers, how it works. So it's very helpful. And I just have a question, you have shown us using the glass beads, and then you use the polystyrene beads. So could you give us more idea about what kind of beads property, transparency, flexibility, or with that index will help to vary this force?
Yes, I mean, so we do the calculations. We do the Maxwell calculations, the finite difference time domain, T-matrix stuff to calculate what the forces and torques are. I mean, the refractive index there has to be a significant difference, but if you go too high, you get other effects that you don't want. So you have to be refractive index of 1.5, 1.6. So water at 1.3, say. So you choose the material. You want it as transparent as possible because you don't want scattering, because that's going to put forces on that that you're not controlling.
And how about the surface property, charged or not charged, or whatever?
I mean, if the probe is away from the sample, it probably doesn't matter. I mean, so we're using. I mean, you can use that. You can actually functionalize the surface so that there would be charge on the surface, but
hollow, solid.
You want it solid. Because the force depends on the volume. So so that's why you need bigger beads. If you make smaller beads, you have lower forces. But you can use the surface. You can modify the surface to have properties that will cause interactions. So you could imagine where you have the 3 spheres. you could put antibodies on one of the spheres, and then that, as you were feeling the surface, that would be sticky, because that would be interacting with certain. So you could feel maybe where you had certain proteins by the fact it was sticky in those areas.
And also now the particle synthesis, you can synthesize the particle with different components. So you have two components, polystyrene versus glass, for example. So they may have different properties. So whether this kind of property would be helpful to you.
Yeah, so we haven't done much with polystyrene, mostly glass. Some of them are polystyrene, but then this polymer, which is proprietary, so we don't actually know what it is. For this two-photon polymerization, fortunately has the right refractive index. We think it's SU8, so it has the refractive index in the right range.
Perhaps I can just ask a last question. You've obviously developed a large number of potentially very powerful tools for examining soft matter of the nanoscale, each one of which might have, the capacity for examining a number of very fundamental or even applied problems. Have the tools themselves been used yet by others to do this?
The contact mode was the first one that we developed, and our spin-out company marketed that for about 18 months. We sold about 10 of those around the world. So the company now focuses on defects review in the silicon fab and other aspects of silicon fab. So yes, that's being used well, it's the next stage. It's being evaluated by the big players at the moment. So for the next generation, for the 22 and 15 nanometer, it's where SEM runs out of steam in terms of causing deposition of contaminants and putting charge. We're pretty unique in doing the vertical probe, I'm afraid. I'd like more people to do it. And of course, holographic tweezers people have done, but not made the AFM from it. But, yeah.
The structure of water is an important problem that people have looked at for many, many years. But with the ability to detect a change in viscosity of the probe tip, have you speculated at all on what the structure of water might be under this condition?
It's very interesting. I mean, we've looked at various surfaces to try and look at the water structure. Obviously, you start with a hydrophilic, then you look at a hydrophobic, and then we've looked at sugars, because sugars are quite amazing in that they're all very similar structures, but they behave very differently. From cellulose through to glucose, or you have a very different insoluble or soluble property to it. So we've been looking on situations where we can make all the hydrogen bonding on one side and then turn it over and look at, and we can easily detect which side of the sugar we're looking at by the change in resonant frequency of the probe. So, but I don't understand it all. I mean, it's a very rich area, I think.
Well, no other questions. I think we've heard a fantastic lecture today, so please join me in thanking Professor Miles for his brilliant report.
Thank you.
Great lecture.
Thank you.